Hello, everyone. If I have not met you yet, my name is Lara Milligan, and I am the Natural Resources Agent. I work for IFAS Extension in Pinellas County. I am not based in the main office, though. I'm located up at Brooker Creek Preserve, so feel free to come say hi if your boss lets you do so. But I just wanted to give a quick rundown. You may have heard about doing snake research or collecting dead snakes, depending how long you've been with the department. So I just wanted to give a quick rundown, refresher, reminder, or if this is brand new to you, kind of the overview of what the heck we are doing and why. So again, this is who I am. I've been in this role since 2012. So going on 12 years if I did my math right, which is always questionable. But basically, at Brooker Creek Preserve, if you've been here a while, we historically were known for our pygmy rattlesnakes. It was like, if you want to see one, you go to Brooker Creek and it's guaranteed. So on the left, this is just one example. This is from October of 2016 from one hike around our education center trail, which is only about 3/4 of a mile. We saw 7 pygmy rattlesnakes just on one little loop, many of which were mating. And kind of fast forward, we honestly recently saw one pair mating, which we haven't seen in years, but more likely than not, we're not seeing any at all. And if we do, like this is one example, we had a visitor say, hey, they ran into a pygmy rattlesnake down this trail. And we were excited because we hadn't seen one in forever. And we hiked down there and it was still there, which we thought was a little odd because it was in the middle of the trail. And it just looked like we thought it was dead. It ended up not being, but it anyway kind of just looked sickly. And so it kind of prompted us to start questioning what's happening with our pygmy rattlesnakes. Fast forward, I did some reaching out to people, reach out to the Rattlesnake Conservancy. They put me in touch with a PhD student in Florida who is studying, ironically, the decline of pygmy rattlesnakes. So it's like, wow, I'm not the only one. Like, this is real. So basically, I got in touch with the researcher. I said, how can I help you? And this particular decline is being associated with a new invasive parasite called RO. That's short for its very hard to say scientific name, or snake lungworm is sometimes what it's referred to as. But basically, they hadn't documented it in Pinellas County. And so I said, how can we help? Like, what can we do? So in talking with the Parks Department, I said, listen, we have, 20 plus parks and preserves and, close to 90 rangers that are out there all the time. So is there any way they could help us to collect roadkill snakes for this research? And so this is part of a larger effort, like actually goes beyond Florida, but it's called the Snake Lungworm Alliance and Monitoring, which we short for SLAM. So that's kind of the bigger partnership. And then locally, we're just really partnering between us with Extension and then the Parks Department. So we are aiding in the collection of these roadkill snakes, and then basically we get the snakes to the research team, who then does all of the dissection and molecular analysis to confirm the particular species of pentastomes that are present, if any at all. So we are currently, you can see on the map on the left is the most recent. This is actually unpublished data. This map is about to be published, but this is the spread so far of the invasive parasite that's impacting not only pygmy rattlesnakes, but all of our native snake species. And I'll get more into that in a second. But you can see it's kind of been concentrated more like in South and Central Florida. And then most recently, we've been finding it elsewhere out towards the coast. And then you can see the furthest out into the panhandle, which is obviously concerning because then we're getting into neighboring states as well. So this is really helping not only to document the impact and understand it here in Pinellas, but beyond as well. So the snake lungworm, the kind of common name for it, they're referred to as pentastomes. And I know this is a little graphic, but coming out of the snake's mouth on the left are the actual pentastomes. And so while we refer to it as a snake lungworm, that's just kind of, again, a general term, but they are technically crustaceans. So we say they are worm-like crustaceans. You can see a picture of one in a vial down on the bottom right as well. And basically they parasitize the lungs, not only of snakes, but they can also of mammals, just broadly the group of pentastomes. and they rely on the host's blood. That's how they survive and thrive and reproduce. That is like their sustenance and food source. And basically what we are seeing is severe infections. So basically having lots of these pentostomes in their lungs are kind of leading to having sublethal to lethal impacts. What we don't have definitively yet is to say that these pentostomes, the particular one we are studying, is the cause for this decline. It's kind of assumed, that's like the hypothesis, if you will, right now. And so we are collecting the data to prove or disprove that. The pentostomes are actually believed to be a spillover from the Burmese pythons, which, as you may be familiar, are more concentrated in South Florida and like Southeast Florida. And so the pentostomes are native to them where they are native to. And so now we are finding them in our native snakes. It's been documented. This is the same map I showed you before. So we're now at 28 counties. We have 67 in Florida, so lots more research still to be done. But we are adding new counties regularly, and it just, if you're familiar with academia, it just takes a while to write everything up and get it accepted into journals and things like that. So it's a work in progress, quite literally, and the particular PhD student that we are working with has no plans of stopping this research, so this is going to be ongoing. If anybody asks you, we plan to collect snakes kind of indefinitely, at least for the next 4 years into the future. So the picture here, you can see on the top left, the Rallatalia orientalis, if I even said that correctly, which is why we call it RO. That's the scientific name for the particular pentostome that we are studying right now. And the snakes are kind of the final definitive host, and I'll show you the life cycle next. But once the snake dies, whether it's from the pentostomes or not, the pentostomes will actually emerge out of the snake's mouth. So definitely if you find a roadkill snake, take a look at its head area, and if you see any weird worm-like things coming out of its mouth, I would encourage you, A, to collect them, but B, at least at a minimum, snap a picture, and I'll get more into the collection process in a minute. So this is the life cycle on the left. So you can see kind of number one here starts with, it's like a picture of a roach, and then we have different species here from lizards, frogs, and smaller mammals. And then kind of the last phase of this life cycle is in the definitive or final host, which is our native snakes. So this is just, again, putting it more into actual pictures. So coprophagus just means that you eat your poop. So rabbits are known for eating their own poop. That's just the fancy term for it. And so basically snakes that are infected with the pentostomes, the eggs from them will be excreted through the snake, and then things like roaches will eat the snake feces, and this kind of is what gets the cycle going. So now the roaches are infected with eggs of the invasive pentostome. Inside, so then let's fast forward, I'm getting ahead of myself. So then the lizards or other creatures like frogs that might ingest something like a roach, so that's kind of the next phase. So they'll eat the roach, so now they have the eggs in them, which have grown and matured, or will grow, mature, and become kind of the larval phase of the pentostome inside of them. There's no documented impacts right now in kind of what we call the intermediate phase of this life cycle. So they're just kind of like carrying, that's why they're the intermediate host. Then if a snake eats the infected lizard or frog or smaller mammal that happens to have the pentostomes of the larval stage in them, they will go into the snake and, I thought I had the next slide, but basically that's when they molt and get into their final reproductive form inside the snake. They will go up into the lungs, bore into the lining of the lungs, and that's where they kind of suck the blood to sustain themselves. And that is what we are studying, is what is the impacts of that to our native snake species. So how can you help? Here comes the fun part. Well, maybe, depending who you are. So I have put together a document, which basically this video is really like a more in-depth version of that document. But if you want to go back and reference, if you're more of like a learn by reading, I will have a link to that document at the end, as well as hopefully you have a printout, or I will have that emailed to you that kind of outlines the process. But then once you're familiar with the process for collecting, you will go out and collect roadkill specimens. Of course, it's opportunistic. It's not something you can like spend time going out and seeking or looking for. It's just kind of as we find them, we are encouraging and asking you to collect them. And then we, at certain times of the year, when I know the researcher will be in town, I will reach out and try and coordinate to get all the snakes in one pickup location for the researcher. So specifically, we are collecting roadkill snakes of any species, but the key is we really want them to be in relatively good condition. So as the researcher told me, ideally still 3D, not flattened into 2D. So if they're totally mutilated, we really honestly, the sample's not going to be helpful for analysis or anything. So that's just kind of like let nature recycle that and, do its business. Somebody will take advantage of that. Otherwise, if it is in relatively good condition, we are asking them to be collected. So before you do the collection, there's a couple things that you will need. One is a Ziploc bag to collect your specimen in. Second thing will be a permanent marker to document your finding on your bag. And then preferably to have some type of protection on you in the form of gloves, or some people I know have used like turn the baggie inside out, maybe like you would do to pick up your dog poo. And then you can kind of flip it to keep your hand protected from the snake that way. Another option, if you're not comfortable using your hands, which I totally get, I personally would not, if you have a snake hook available or tongs, or even I know most of the parks have trash pickers and you can utilize those to pick up the snakes and place them in your bag. Then place the baggie and the specimen into your freezer, and then there will be data collection sheets. You should have access to these if you want to document on there. I'm mostly the one that's keeping track of that. However, if you at your own site want to document that, that is available to you. And all of the data that you're going to be writing on your baggie is what I transfer onto my larger data sheet. So, okay, you come across a dead snake. Now what? So you gather all of those materials. First and foremost, depending where you are, right, we want to ensure your safety. So if you're in a high traffic area, whether it's bikers or cars, just make sure that it's safe before you go to collect your specimen. And then we just really encourage, especially if it's a venomous species, or if you're unsure, if you're like, I don't know what snake this is, we just encourage you to use extra caution because they can still strike. Sometimes there's kind of like this last nerves remaining in them. So just make sure, the specimen's really truly dead, or just in general, use extra caution with venomous species, and definitely not touching directly with our hands with venomous species. Then you will collect the snake. Like I said, you can use snake hooks, tongs, trash picker, put it in your baggie, seal it. Ideally, try and get as much air out of the baggie as you can, because it will be going into the freezer. And then what we want to document, really, the more the better. But at a minimum, we would like to know your name, who is collecting it, the date in which it was collected. We don't necessarily need the time, although, again, any additional information is helpful. And then if you can get a GPS coordinate, that's more preferred. However, if you just have generally the name of your park, that's also a-okay if you don't want to stress about the GPS. So those are the main things. If you know the species of snake, again, you can add it on there as well, but the snake researcher will definitely be able to ID your snake for you. And then again, you're placing that in your designated freezer. I know not all people are like us at Brooker, where they don't mind if their food is mixed with snakes. Thank goodness we're not inspected by the health department, but I'm working on getting a few new freezers out to some of you. So definitely let me know if you're still in need of a freezer. And then that's it. Repeat. And they can stay in the freezer, as long as they need to. It won't impact the specimen at all. And then again, I will reach out once it's time to kind of gather them all in one spot for the researcher to pick up. So I appreciate your time. I really appreciate your contributions to this effort. If you want to participate, and again, refer to your chief rangers on what your roles and responsibilities are in this. I know that this has been supported by higher admin, your involvement in it. So there's definitely no hesitation from the top. If you do want to learn more, you can either pause the video here. There's lots of resources. So first is the document that I mentioned before with the standard operating procedure for collecting the snakes, and I will again get that to you. I do have a podcast called Naturally Florida, and we did a whole episode on the invasive pentastome. So if you kind of want, I went over a lot of it here now, but if you want more, just a quick refresher, if you like to listen on 2-speed or whatever like me, you can scan that QR code. And then we have 2 other videos. One is by, actually they're both involve the actual researcher we were working with, Jenna Palmasano. And so the first is a program that she did here at Brooker Creek Preserve that kind of talks about the research that she's doing. So that's available. And then she also did a dissection if you just are curious on like what happens to your frozen roadkill snakes once she gets a hold of them. This is just the dissection part, and then they do a more formal analysis at the lab, and including molecular testing, because anyway, it gets very complicated in terms of verifying that this is the particular pentastome in question. So those are additional resources if you want to learn more. And of course, I am happy to answer any questions that you have. I probably should have put my email on here, but you can find me in the parks directory. Just search for Lara Milligan, just Milligan at pinellas.gov. So with that, I appreciate your time and again, your contributions to this effort, and happy to answer any questions that you guys have.